Perfect. Well, yeah, thank you for having me. My name is Kavi Ganalan, and I'm excited to talk to you today about the DataJoint tools we have and to build DataJoint workflows for neurophysiology. And I'll be building off of the talk that Dimitri gave yesterday. And later this afternoon, my colleague David will be giving a workshop to show you how to get up and running with DataJoint. So this is the outline of the talk. So we'll be going through the problem statement that we're trying to address, and then talking about what is DataJoint, the company, the open source tools. What does a typical neuroscience experiment look like? And then how do you engineer a scientific workflow with DataJoint to manage all the data from a typical neuroscience experiment? And then what are some DataJoint tools that, like DataJoint Elements, that we've developed to help facilitate developing scientific workflows for neurophysiology? And then summarize kind of the suite of the tools that we have. So the problem statement, it was well articulated yesterday during a number of the talks, whereby in neuroscience, the experiments are getting more and more complex. The data are getting more and more large. And so this is just an example of microelectrode recordings from ePhys, streaming in gigabytes of data for a given experimental session. And then alongside this, you could have different modalities of acquisitions. So let's say you have some sort of behavioral data that you're then analyzing further with the deep lab cut. And so the amount of data is very large, and piecing these different packages together, or the outputs from these different packages together, is a challenging process. So what we're trying to do is help scientists, or rather scientists typically develop custom scientific workflows with limited resources. And these are PhD students, postdoctoral fellows who have to develop these custom workflows. And every single time, given that they have to develop custom workflows, this really slows down scientific discoveries and it hinders reproducible science. From my own personal experience, during my PhD, it took 2, 3, 4 years to develop the custom scientific workflow to answer the scientific hypotheses that we have, albeit in a different area of neuroscience than what I'm presenting on today, but it still took that order of magnitude to develop our scientific workflow. And what I mean by hinders reproducible science is that if everyone's developing their own custom scientific workflow, then for a reader of a manuscript to go through the methods section, to possibly find the GitHub repo with the source code, to possibly then find the separate repo with the raw or processed data, and then to piece all those things together to actually reproduce a study is very challenging. And so these are the types of problems that we're trying to address here at DataJoint. And so what is DataJoint? It's a set of open source software tools. You can find more about it at datajoint.org. It was actually started by our CEO, Dimitri, during his PhD back in 2009, and has grown. And we now have a team of engineers who further develop it and develop the suite of tools we have around that DataJoint framework, which I'll talk more about today. DataJoint is also our company name. You can find more about us at datajoint.com, where we help academic scientists, much like y'all on the call today, to implement a DataJoint scientific workflow for your scientific needs. And I would say that every engineer and scientist at DataJoint works on the open source side of things in some capacity or another. And so there's this real nice synergism between having this open source set of tools, helping academic scientists implement them, and then taking the funds from that and reinvesting it into effectively engineering time to further develop our open source tools. And so at DataJoint, we're a group of largely engineers and scientists, and we're driven by this core mission to enable research teams to solve the most complex problems in their field by bringing scientific rigor and clarity to big data and computations. As an aside, you'll notice that actually the word neuroscience is not in here, because our DataJoint framework can actually be applied to really any scientific workflow, which is really nice that it's extensible in that way. We have focused on neuroscience entirely since our inception of the company, because the co-founders and everyone since then are neuroscientists by training. And we're also guided by 4 key values. So scientific integrity, what I mean by that is we develop tools that are scientifically rigorous, that can allow for reproducible findings, and you can then draw valid conclusions from these findings. Innovation, we really seek to understand what the community needs are and then innovate new technologies to adapt to those needs. Approachability, in which we support research groups of different sizes and whose members maybe have diverse backgrounds, resources, or skill levels. And the community, we seek to understand what are the other tools that are out there, what are the other communities that are around those tools, that support those tools, and how can we work with them so that we all kind of grow together to help the neuroscience community in general. So that's what DataJoint is. And so now how do we apply that to a neuroscience? So let's talk about a typical or a potential neuroscience experiment. So it could start with an animal, the subject, and then you would perform some sort of experimental session, maybe daily, maybe every other day or so. And then let's say you acquire some sort of modality like behavioral data. And then next, you would do some sort of behavioral analysis of that data, let's say with deep lab cut to track the animal's movements. And then we're all neuroscientists, so we want to record from neurons in some way. So let's say we use some sort of modality to record electrical activity from neurons. And we want to process that data. And so we have to use some sort of parameters. Let's say we want to detect the spikes. When the spikes occur, the action potentials occur. And so we have to store and save the parameters we want to use for our experiments, for our analysis rather. And so then we have a spike detection step where we detect when action potentials are occurring. And then we want to actually answer a scientific hypothesis. So we want to do some sort of statistical analyses, population-based, so that we can answer our question in a scientifically rigorous way. So this is what a neuroscience experiment might look like, and I'll use this as kind of the template for the rest of the talk to kind of show you how DataJoint works with this. So we're going to engineer a scientific workflow with this framework in mind. So each one of these shapes here represents a step in your scientific workflow. And the real challenge that we're trying to address is passing data between these different steps. And each step may represent a different analysis package or acquisition package. And packages change over time. And so that's what we're trying to develop a framework to handle that. And so the first step in kind of engineering scientific workflows, seeing the data that's being acquired. So for metadata for the animal and the experimental session, maybe you store that in some sort of lab notebook. It could be a physical notebook, it could be an Excel sheet, it could be a Google Doc, something like that. For the behavioral data, it's a movie, so it's some sort of AVI file that you're acquiring. For the neuronal traces, the voltage traces, you're going to be acquiring some sort of file. Maybe it's saved as a binary file, .bin. The behavioral analysis, maybe it's done in MATLAB, so it's saved as a .mat file. The spike detection parameters, that could be some sort of notebook or some sort of sticky note or something like that. I know we've all kind of been in those shoes before. And then maybe the spike detection algorithm is in Python, is Python package. So you save that data as a NumPy array, NPY, and the statistical analyses is maybe also done in Python. I know during my PhD, this is how I kind of operated. I had basically a Excel sheet that stored all my metadata and then separate files, and I built the scientific workflow around passing data between these different steps, reading in and writing out these different file formats. But one way of addressing this is actually through using databases, and that's where our tool that we've developed, DataJoint, comes in, is handling how do you pass data back and forth between these different steps of your scientific workflow. And DataJoint is a framework. It works in common scientific computing languages to create these workflows, such as MATLAB and Python. So you can think of technically DataJoint as a Python or MATLAB package that you can import. And then once you import it, you'll have the power of DataJoint within your Python or MATLAB script. So if you're familiar with Python, you would just type in import DataJoint once it's installed, and you would have the power of DataJoint there, right there in your script. So let's talk a little bit more about the DataJoint framework. It allows you to model each one of these steps in your scientific workflow. So you can think of these steps kind of independently of each other. So the different steps are possibly different algorithms that are run or different acquisitions systems that are acquiring data. So you have to account for that, these different packages. And then you also, as I mentioned, you also have to take into account how data flows between these different steps. And I'll be talking a little bit more about this in the next few slides, but it's important to highlight that with DataJoint, when you model this scientific workflow, each of these nodes in this graph represents a table in a database and a block of code that defines how data is entered into that table in a database. So let's take an example of how to create a table with DataJoint. And let's look at it in Python, for example. So let's look at the neuron table. And as I mentioned, each of these nodes is represented by a block of code in DataJoint. And so here, what I'm showing you is that block of code in Python, and that block of code then defines a table in a database. So let's walk through this block of code a little bit more. I'll still keep it pretty high level. During the workshop, David will go through in more details what each line of code means. So that at schema at the very top, it's a Python decorator. For those familiar with Python, it links to the database of that you've declared. And then the next line, class neuron, and then in parentheses, DJ, meaning DataJoint.imported, it defines the name of the table that you want to define and its type. So in this case, it's an imported table. So I should back up and give you a little bit of a story in terms of like this data. So let's say for this example, we're acquiring some sort of electrical traces of neurons. And the data from one experimental session is stored for multiple neurons in the same file. So that's the data that we're trying to insert into this table using this block of code. And then in the next section, we have the definition, which defines the table column names and then how this table is dependent on any other upstream tables. And so when you define that code and those table column names, that declares it within the table in the database. And each table has some sort of unique identifiers so that you can identify an entry in that table. So in this case, for the neuron table, the subject ID, the session date time, and the neuron ID all uniquely will define that specific neuron's trace for that experimental session. And then in the next block of code, it's the make function. So if you end up using DataJoint quite often, you'll learn and hear about the make function quite often. It's one of the powers of DataJoint. That's where actually all the table computations happen. So if you want to run an algorithm, run a spike detection algorithm, that's where you would put it, is within the make function for that table. So I'll just highlight what this block of code does is that it parses a file for an experimental session, looks through, finds the voltage traces for the different neurons that are saved for that experimental session, and then it inserts it into the database. So in this case, we have two neurons, and their voltage activity is saved in the database. I've just shown it here as a ... but you can imagine that's a voltage trace. So now that we've defined one of our DataJoint tables in our scientific workflow, let's talk about how data actually passes between the different steps of your scientific workflow. And I'll just pause to say that this is one key slide I would like you to take away from today. is how each node within your DataJoint scientific workflow is represented from the software side of things. So it's represented by a block of code as well as by a table in a relational database. So now how does data actually flow through this workflow, assuming that we've defined that block of code for every single one of these nodes? So as I mentioned, each one of these nodes represents a table in a relational database. And so the first step would be to manually insert data into the most upstream table, the topmost table, so the animal. So we're going to insert data into this animal table. The table is defined by the columns subject ID, birth date, and gender. So we're inserting subject ID, let's say subject is numbered 1, the birth date, and the gender. And to pull back a little bit of the curtain behind how DataJoint does this is it's using this command. So in Python, you would say for the animal table, you will insert one entry into the table with the subject ID set to 1, the birth date, and the gender set as well. And I will reinforce that the subject ID in this case is the unique identifier for this table. So no two subjects can be entered, no two subjects with the same subject ID can be entered into this table. And so next we want to insert into the experimental session table. So let's say we have another subject, or sorry, the same subject, subject 1, for an experimental session date time of June 1st at 6 a.m. And there's no experimental notes for this case. So in this case, in order to do this, you do session.insert, subject 1, session date time, and the session notes. So next, we want to insert into the neuron table. This is the table that I showed you had a block of code associated with it. So this table is defined by the columns of subject ID, session date time, neuron ID, and activity. And so because we've defined that block of code for this neuron table, and because secondly, we've defined up here in the session table that there's a subject ID and a given experimental session, DataJoint can actually look at that experimental session and run this block of code for that experimental session, just with this one simple command. So you say neuron.populate. What it'll do is say, okay, what experimental sessions have not been run yet? So in this case, subject 1 and session date time have not been run yet. And it'll run that block of code within that make function to insert the data into the neuron table. And that's what happens here. So we insert the subject 1, session date time, the same. And then there was 2 neurons in that one text file that was, or binary file that was saved. And the voltage traces, which are, let's say, an array, were saved into the activity column here, which I'm just denoting again as a ... but it would be an array of values. So this is the real power of DataJoint, is that it finds all the entries that haven't been processed yet, and it processes it for that table that you say populate. So now let's continue on to our next table. So the spike detection parameters could be a manual table as well. So you manually insert the parameters you want to use for your spike detection algorithm. And I'll skip over the steps of inserting that data, but you can imagine it's much like the animal and the session tables. And then next we want to run the other tables. And these other tables, we didn't go into the details of them, but the behavior table, the spikes table, the behavior analysis table, and the statistics table are all tables, much like the neuron table, where we defined what the table columns would be, as well as the make function, where the computations, the algorithms that are run will be and what they are. And so again, just like the neuron table, you run spikes.populate, behavior.populate, and so on to process that data for that experimental session. So this is another key takeaway, is that with DataJoint, you can run these automated computations. So you insert metadata for your experimental session into the session table, and then you maybe every night you have your computer run a job where it says spikes.populate, neuron.populate, behavior.populate, and that'll process the data through your workflow and insert all that data into your DataJoint tables. And more technically speaking, this can be done as a cron job or however maybe you want to set that up, but that can be done on a regular basis. So you don't have to worry about it. Once you set up your DataJoint workflow with all the computations and algorithms within your make function, and you run the populate on your experiment for your experimental sessions, it'll populate all that data and process all that data into your database directly. So now that we've defined our scientific workflow, our DataJoint scientific workflow, and run through an experimental session through our DataJoint workflow, let's talk about how you can actually use kind of DataJoint Python languages or MATLAB languages to actually search for data that you're interested in. So let's go back to our neuron table and talk about how you can search for data. So let's say we've actually been doing a lot of experimental sessions. So we have, in this case, 3 subjects, but you can imagine there's hundreds of subjects, hundreds of experimental sessions, thousands of experimental sessions, and you want to find one specific neuron to plot. How would you actually do that? Well, using DataJoint syntax, you would display the table name, so neuron in this case, and then you would use the ampersand, which in DataJoint it's an operator to say we want to select or filter only those tables that meet this criteria. So we want to find the table, we want to find the entries within the neuron table that are subject ID equals 2 for a given session date time and for neuron ID number 1. And so if you run this command in your Python script or your MATLAB script or something similar like to this in your MATLAB script, it will return that entry. And then using one more command, which David will show, it's .fetch, it will download that data onto your, let's say, local machine where you can plot that data for your downstream analysis. So for anybody who knows SQL, this is, for me at least, it's much more intuitive to write these kind of queries, these kind of searches for a database in the DataJoint language as opposed to SQL commands. And so once you have all your data in your database tables, DataJoint really promotes open science and collaboration. It's really a natural extension from your DataJoint workflow. And so, as I mentioned, each step in your workflow is a DataJoint table. It's in a database. And using the power of DataJoint, whether it's in Python or MATLAB, you can search for data in the database. And this can be done by you, by a collaborator, by your PI. And this is done while your experiments are being conducted. So not after your study has been published in a journal, but actually while you're still collecting data. So if you need to tweak things, you can analyze your data and then iterate, tweak on your experimental protocols if necessary, or just have further discussions with your collaborators. And so that's the real power. That's one of the additional powers of DataJoint. And I've talked about how to look at data and insert data through Python language, but let's use a GUI as well, because sometimes GUIs are nice to look at data and visualize what's going on. So we have another tool that we released this year, DataJoint LabBook, which is a web app with a REST API on the back end. And you can log into a database, a DataJoint database that you set up. And on this next slide, there are 3 columns. The first column on the left shows the database schema. I know I didn't go over the term schema, but it's where all your tables are housed. The second column are your tables. So in this case, we have a computer table and an employee table, just as an example. And on the rightmost column, once we select a table, it'll show us the contents of that table. So we have here the contents of our table, which is 2 entries. So we can look at the different table columns and the different values there. And then if we had thousands, hundreds of thousands of entries, we'd want to filter to find the exact entry we're interested in. And so using that filter button at the very top, you can say, I want to find the computers that are cost less than $800 and computers with greater memory than 16 gigabytes. And just like we did within Python, we're able to actually insert data into our tables here using this GUI as well. We can update entries. So if we have an entry and we find that there's something wrong with it, or we want to make a modification to it, we can update that entry. And we can also delete entries as well. So that's our graphical interface for our DataJoint database tables. And so that's really the DataJoint framework and a few of the tools we have built on top of the DataJoint framework. And so to kind of summarize, DataJoint is a kind of an open source framework for building scientific workflows. And it's kind of based on these core principles. The first one is sharing live workflows. As I mentioned, it allows for real collaboration while your experiments are being conducted and acquired, and while your data are being processed. What I mean by data structure here is that you can define what each one of your columns are, the data types, and provides a structure for your data, as opposed to just storing it in a bunch of folders and whatnot, or an Excel sheet. And then data integrity, it basically means that when you insert data or when you delete data, that it will delete the entire data set and it won't keep kind of parts of data sets within your databases. Data consistency is important because through DataJoint, it creates one kind of consistent view of your table. So if two people are simultaneously reading from your database, they'll see the exact same things. And multiple people can actually simultaneously write to your database or near simultaneously write to your database. And other people can then see your updates in near real time. And has to have high performance. We're working with gigabytes, terabytes, petabytes of data, and so it has to be extensible to that. And then once you're ready for publishing, I'll talk about some of the tools that we're planning on developing to further promote the FAIR principles as well. So that's DataJoint overall. I do want to talk about DataJoint Elements, which builds on top of DataJoint. So DataJoint Elements are for neurophysiology. We have a preprint that is out this year that talks about DataJoint Elements. So before I get into it, let me talk to you about the problem that DataJoint Elements addresses. So as we talked about, these scientific workflows can be developed. They can actually be customized to your specific needs. So you can set up a neuron table and not the behavior table, or vice versa, or other modalities. And it kind of depends on what your scientific questions are. But that could take some time, and it would mean that every single lab would be using it would be using different workflows in effect. And it would take time because you're really defining what that block of code is that defines each one of your nodes in this graph. And so before I talk to you about the DataJoint Elements that we've developed, I just want to kind of further reinforce some of the advantages of using DataJoint Elements. And what I mean by that are standardized workflows. So DataJoint Elements, we've developed them kind of like standard Python packages where you can install them using pip and import that DataJoint element and use it as if it's a standard package. So standardized packages are beneficial because they kind of reduce the time to implement DataJoint. So as a PhD student, postdoctoral fellow, it'll really streamline that process of starting up with DataJoint. This also allows for a modular workflow design. So let's say you're working with electrophysiological data and behavioral data, but now you want to start with calcium imaging data as well. You can import the calcium imaging element that I'll talk about shortly and have that entire workflow set up for you. And then the next point I wanted to make was that it facilitates collaboration to advance a given workflow for the neuroscience community. So what I mean by that is if you have one workflow that many labs are using as a starting point, everyone can collaborate on that one workflow, on that one starting point to improve it because of their experiences, any bugs they might find or anything like that. And that will help the neuroscience community overall, as opposed to each one of us kind of creating our own scientific workflow individually. And along those same lines, it promotes reproducible science. If you have one workflow that and if you have one workflow that's published on GitHub, like we've done with the DataJoint Elements, anybody can download that workflow and use the parameters that you use for your analysis and reproduce your findings. So we have two DataJoint Elements that are in beta release right now. One is the DataJoint element for two-photon calcium imaging. So calcium imaging, as you all may know, is imaging neuronal signals kind of deep within intact brain tissue. And once you acquire the calcium imaging signals, you have to process them. And there's many different steps to process them. And this is our workflow that we've defined that processes those steps. And this is based on experience working with leading labs all along the world who have done calcium imaging experiments and processed that data with DataJoint workflows. And so as you can imagine, each one of these nodes is defined by a block of code. And so we've already defined that block of code for you. And so instead of having to define each one of those blocks of code, it would amount to approximately 9 lines of code to have this DataJoint workflow up and running for you. So to kind of go through these lines of code individually. So in the first line, you would import DataJoint. And in the next block of code, you would import the different DataJoint elements. So element animal, element lab, and element session all store metadata for your experimental sessions. And then element calcium imaging is the bulk of it, which processes all your calcium imaging data. And so once you've imported those packages, you have to create a version of it on your database that you've connected to. And so you do that with the activate function at the very bottom of this block of code. So lab.activate, subject.activate, that instantiates those tables on a database. And so for those familiar with two-photon calcium imaging, our current element supports acquisition systems for ScanImage and ScanBox, as well as analysis packages from Sweet2P and Kaiman. And so I wanted to highlight 2 points here. One is we're looking to support more and more commonly used packages. So if you have something or know of some package that is of value to the neuroscience community, please do reach out to us. We are working to support more packages. And then 2, this using different analysis packages within the same framework. So for instance, Sweet2P and Kaiman are pretty commonly used to analyze two-photon calcium imaging data. This having one framework that unifies these 2 packages allows you to compare your results from Sweet2P and Kaiman pretty directly for one given experimental session. So that's another power of these DataJoint elements. So this is the DataJoint element for two-photon calcium imaging. We also have a DataJoint element for array electrophysiology, specifically for data that's acquired with the Neuropixels probes. And this element supports acquisition systems like SpikeGLX and OpenEFIS, as well as the analysis package KiloSort. So those are our DataJoint elements that build on top of the DataJoint framework to help neuroscience labs build DataJoint workflows. And these are standardized solutions for analyzing and organizing data into a shared database. They're all open source, as I mentioned. They're hosted on GitHub. And they allow for comparison between different analysis packages within this one same framework. This project is actually funded by the National Institutes of Health. It's a NIH U24 grant. It's called DataJoint Pipelines for Neurophysiology. The principal investigator is actually the CEO, Dimitri, and we're in year 2 of the 5-year grant. And so we've developed, as I mentioned, the beta release for two-photon calcium imaging, as well as array electrophysiology with Neuropixels probes. But this coming year, we're working on developing future elements in beta release. So we're working on miniscope, sensory stimulation, histology to locate the probe trajectories, brain coordinate frameworks, recording behavior with automatic, and then analyzing with automated pose tracking, and then trial-based experiments as well. And we're also looking to add further features to DataJoint elements. So we're just like computations are embedded within the make function of the DataJoint tables, we were trying to embed visualization. So basically every single table should know what type of data is in it and how do you visualize that specific type of neurophysiological data. So that's something we're working on. Synchronization, so what I mean by that is how do you synchronize data between two different modalities? So if you're acquiring behavioral data and calcium imaging data or electrophysiological data, how do you synchronize those data sets together so that you can actually do your scientific analysis? And then we're looking, as we heard about yesterday and we'll hear about today, we're looking to export to NWB, and we're looking to publish to Dandy Archive as well to further promote FAIR principles. And so that is, those are some of the high points of DataJoint, or those are some of the resources of DataJoint Elements. But I do want to talk to you about a few more that I haven't had a chance to talk to you about yet. But all of these can be found on datajoint.org. So let me step through all of the resources we have built around the DataJoint framework. So in the left column, I'll just explain the resource, and on the right column, it'll be the URL. So we have DataJoint for MATLAB and DataJoint for Python. The source code is on GitHub, but you can actually install it via the MATLAB Exchange for MATLAB and via PyPI for Python. DataJoint Playground, it's a really cool environment. It's a Jupyter environment where you can log in using a free DataJoint account and use our DataJoint Jupyter notebooks to understand how this framework works and how do you build an actual workflow, scientific workflow, and to actually work with some data. And we're constantly adding more and more tutorial notebooks, so you'll hopefully see this grow over the next couple of years. I talked to you about our elements, DataJoint Elements, which build on top of DataJoint. And then we have an Elements Early Adopter Program. So as was discussed yesterday, it was one of the big questions is how do we help neuroscientists use these open source tools? Well, one way is we're funded by the NIH, and one of the big pushes for our grant is to help neuroscientists use all of our elements that we've been developing. And so this is what we're calling the Early Adopter Program, where it's entirely funded by the NIH to help scientists like yourselves use these elements that have been developed or the upcoming ones. And this is mutually beneficial because hopefully you would benefit from it, or scientists would benefit from it to actually have a workflow in place that's readily usable. And then we and the greater community would benefit from it because we would use your expertise as scientists to validate the results coming out of these elements as well. So please do check out datajoint.com/adopt if you're interested in working with us. And then another cool initiative that kind of just been released in the past like a week or two or three is DataJoint Awesome Datasets. So as was mentioned yesterday, there's a microns dataset that was done in collaboration between scientists at the Allen Brain Institute, or Allen Institute for Brain Science, Princeton University, and Baylor College of Medicine. And what they did was they studied a cubic millimeter of brain tissue from the mouse visual cortex and with many different techniques. And this data set is actually like processed and it's now hundreds of gigabytes of data, but it was all done within the DataJoint framework. And so what we've done is taken that open source data and put it into effectively a Jupyter environment where you can work with that data. So right now it's on microns.datajoint.io. We're planning on selecting more published data sets that have used DataJoint to host for free access to the public. So please do check it out. And I will say that Dimitri has told me that he's worked on this data set before, and there are lots of scientific findings that he just hadn't had a chance to publish on. So please do work with this data and see if you can find some cool scientific findings from this data. And then we have DataJoint LabBook, which is our graphical interface for DataJoint tables, and then Ferris, which is our REST API on the back end. And then lastly, we're here to support the community to use DataJoint. And so we're here to answer any questions on DataJoint Slack. And then we would ask that you would post any technical questions about DataJoint on Stack Overflow with a DataJoint tag so that we can, so that it's searchable by Google and anyone who could benefit from the answers to those questions. So our team is comprised of a number of neuroscientists who work with academic scientists to help implement DataJoint frameworks for their scientific experiments, and who've also been developing the DataJoint Elements for neurophysiology. We have a team of software engineers and data systems engineers who develop DataJoint, Python, MATLAB, the DataJoint playground infrastructure, the awesome datasets infrastructure, and many more things, as well as our business operations personnel, our co-founders, and then our scientific steering group for our NIH U24 grant, which focuses on the DataJoint Elements as well. And I'll take this moment to just say we're growing. We have lots of different initiatives that we're working on. And so if you're interested in kind of positions of software engineering, or if you know a neuroscientist with software engineering expertise, please do reach out to us individually, or we have some postings on LinkedIn as well. And then lastly, our workshop will be today at 2 p.m. Later this afternoon. And then for the workshop, it'll be like a live interactive workshop. And we want people to work with our Jupyter notebooks that we've developed for this. So the materials are available at nyu2021.datajoint.io. And so you can use it during the workshop, you can use it after the workshop. And just know that you'll have to create a free DataJoint account prior to logging in. And the workshop will be outlining how to create a DataJoint workflow, how to interact with the workflow and search and download data. I kind of scratched the surface on it today, but there'll be much more time to kind of go into those details. And then as well as how do you automate computations with DataJoint. I talked about how that's the power of the make function and the populate method, but how do you actually develop that? And David will be talking about that in further detail. So with that, I would just like to say thank you. And if you have any questions, I'm here to answer them.